Eros is absolutely one of the most incisive and important contributors to our understanding of chromosome end protection. And so how telomeres engage DNA damage response factors, but then also really control them. And he's made huge contributions. He did his PhD in Milan with Christian Helen, who was at that time in Milan at the IFOM, working on cell cycle control, E2F, P53, stuff like that. Then he went to the Rockefeller and worked in Tizio de Longa's lab as a postdoc and was really kind of a legendary postdoc there. He showed how ATM and ATR signaling pathways are independently inhibited at telomeres, the consequences of telomere deprotection in post-mitotic versus actively growing cells. You know, when do you even need telomere protection if you're not undergoing mitosis anymore? Then he went to start his own lab at the Scripps and was there for about a decade and made some huge discoveries about how TRF2 prevents end-to-end fusions, the mechanisms by which it does that. He identified a new telomere length regulator, TZAP, which turns out to be really important in telomere length homeostasis. He's worked on POT1 and what it's really doing at chromosome ends. And then in maybe like 6 or 7 years ago, the National Cancer Institute accomplished what I'd say is a great coup and recruited Eros away from the Scripps to Bethesda. And we were lucky enough, my lab and me, so this was a major drawback of leaving the NIH, was to miss Eros and his lab. We had joint lab meetings and lots of interactions. And so that was one of the great pleasures of being at the NIH. We learned a lot from him. In recent years, he's done a lot of work on mechanisms of chromosome end protection that differ in stem cells versus differentiated cells. And of course, this work is very important for understanding cancer and stuff like that. So I think there's a lot of interplay between work that Eros is doing and things going on our campus. So thank you, Eros, for coming. And I'd like to present you with an award today as a cancer warrior. Woo!
Oh, wow. Thank you very much. Thank you, Julie. Can you hear me? Does this, okay. So thank you very much. And I think you did really well at recruiting Julie. So I miss, we lost, I mean, I personally miss a lot the interaction that I had with Julie. So it's a pleasure to be here today, see Julie, her lab, and everybody. So today I'm going to tell you 2 main stories, and both of them are going to be unpublished. So, but before that, I have a little bit of introduction on telomeres. So please do stop me at any point if my accent is too strong and you don't understand what I'm saying, or just if I'm not using enough introduction for the telomere part. So please do feel free to stop me anytime. Okay, so let me just remind you, and again, having Joy here, having Julie here, you don't really need a lot of introduction on telomeres, but just why do we need telomeres? The basic, every single organism that have a linear chromosome needs telomeres, and telomeres have 2 functions. So the first one is to deal with the so-called end replication problem. Let me see if I can do, wow, this is, okay. And then replication problem is basically the inability of any DNA polymerase to fully replicate a linear template. So no matter how close you put the last Okazaki fragment, you're going to have a little portion of the telomere of the substrate that is lost. And clearly this over multiple rounds of replication will eventually lead to loss of key genetic information. So you need a way to deal with this. And the most prevalent way that we do, and mammals do it, is using telomerase. So telomerase is an enzyme that recognizes chromosome ends and adds new repeats at the very end. The discovery of telomerase was recognized to be a very important milestone from the Nobel Prize Committee. So this is one way, it's not the only way, but this is one way in which you can deal with the end replication problem. And the second problem connected with telomerase is more pressing. So while telomerase is not an essential gene, you can get rid of telomerase and a mouse is completely fine for multiple generations. The second problem is really, really essential, and it deals with every single cell of our body at all the time. And it has to do with DNA damage. So as you all know, our cells are really sophisticated in recognizing DNA damage and fixing the problem right away. And this is great. That's why how we can maintain genome stability. But this is a problem when you think about the very end of chromosomes, because those are basically double-strand breaks, and they could elicit a DNA damage response that will lead to problems. And the way all organisms solve the problem is by recruiting specific proteins that bind to the telomeres and deal with the different side of DNA damage. And in the case of this is true for all mammals, there is this protein complex that bind to TTHG, which is a sequence in mammals. And you can think about this protein complex like a Swiss knife, where basically each component can do very specific tasks. So you have ways to suppress different aspects of the DNA damage response. Okay, so all of this is good, it's nice, and it's clearly important from a basic biology point of view, but there is also very important consequence for cancer development. So the first one, which is the most obvious in many ways, is the fact that, oops, sorry, let's get used to this. The fact that since every time a cell divides, a little portion of the DNA is lost, and this eventually will lead to cell death, any successful human cancer found a way to deal with this problem by counteracting telomere shortening. So for this reason, Annan and Weinberg recognized telomerase and ways to deal with telomere elongation as one of the key steps for tumor genesis. But also the other aspect of telomere biology, so the end protection, is also very important, because if you have problems maintaining chromosome stability in that telomeres, this will lead to aberrant end-to-end chromosomal fusion. I don't know if you see this, but basically these three chromosomes that are stuck together in these yellow parts, which are telomeres, these are end-to-end chromosomal fusion. So what happened in these cells, there was loss of protection of ends. The DNA damage machinery got very excited, and recognized this as a site of DNA damage and fixed it by engaging in fusion. And this eventually will lead to genome instability when cells try to divide. Okay, so this is all the introduction I'm going to give you. So again, if there is something that I missed or you don't recognize in my next slide, please interrupt me. So the two stories that I'm going to tell you have to do with these two basic mechanisms. So the first one will have to do with how cells deal with telomere dysfunction. And the reason why I'm particularly interested in this is that if everything was perfect, so if every time a tumor arises and the telomeres start to get shorter and eventually they become very short that they cannot protect anymore, we wouldn't have cancer, because at this point, when telomeres are too short, they would elicit a DNA damage response and cells would die, and then there would be no cancer. So clearly this doesn't happen. There's ways in which cells can bypass this problem. So I'm really interested in understanding what are the factors that can modulate the response in which telomere dysfunction is sensed and cells respond to this. And we did a lot of work in vivo, and in vivo, we found that in certain, especially in the brain, when we messed with telomere protection, some cells responded by dying, some cells didn't care, some cells undergo senescence. So there was clearly a very different response. And at that point, I thought that maybe differentiation was a key determinant on the way the cells responded. So, and sorry, I'm going to skip this slide and go directly to the meat of this, which is, so when I started the lab, Marta, that was the first postdoc that joined me when I was at NIH, decided to use a very simple approach to study this project, which was to create an in vitro system where we could have cells that could differentiate in anything we wanted to and see whether there was a difference in the response to telomere dysfunction. So for this project, what we do, we take cell lines that have conditional allele for TRF2, so we can delete TRF2 in a specific manner. And Marta used two cell types. So one of them is mouse embryonic stem cells, which are pluripotent. And then she differentiated exactly from the same cell, some cells that we call fibroblast-like cells, because we don't know what they are, but they are differentiated. So Marta can, using tamoxifen, delete TRF2 because there is a Cree, an inducible Cree that can delete TRF2, deleting these two alleles, and this will lead to a strong telomere deprotection. And we know this because we've been studying TRF2 for a long time. So what happens when you delete TRF2 in somatic cells is quite spectacular. The first time I saw this, I fell in love with telomeres. And in this picture here, you have a differentiated cell. Sorry about this. I will get the hang of it eventually, in which TRF2 is deleted. In a single cell cycle, all the chromosomes are engaged in a single multi-chromosome, basically spaghetti-like, if you want, where every single chromosome is fused together at these yellow dots, which are telomeres. So we deleted TRF2, and the loss protection, the non-homogeneous joining led to end-to-end chromosome fusion. This works in fibroblasts, and it works in every single cell type we ever tested. So the surprise here was one of these experiments that the result was almost too good to be true. It took me a while to be convinced of this result. When we did the same in embryonic stem cells that are, again, genetically identical to these guys, nothing really happened. So the cells slowed down a little bit, but there was no telomere fusions. The cells were viable. We could expand these cells for multiple generations, and they were completely fine. So telling us that, A, we were right. So differentiation does indeed affect the way cells respond to telomere protection, but also telling us that there was something fundamental that we didn't understand about the way cells perceive telomere dysfunction. So we published this back to back, and Simon Bolton had a very similar story. Both of the papers are published a few years ago. So I'm not gonna give you all the details of that story, but I'm just gonna tell you what was our conclusion, which again was quite similar to what Simon presented. So in somatic cells, we understand really well the pathway of TRF2 deletion. You get rid of TRF2, you get activation of ATM. Activation of ATM localizes a bunch of DNA damage protein that then lead to non-homogeneous joining, fusion of chromosomes. So this is a very simple and linear pathway. Somehow in embryonic stem cells, this doesn't happen. And the activation of ATM seems really, so there is some residual activation, but very, very little. We don't understand why. And what we proposed in our paper was the induction of really early embryogenic genes, so so-called 2C genes, such as DISCAN4, were maybe involved in this process. And so this dampening of the response was the reason why these cells didn't fuse and could somehow survive. Simon has a slightly different version of it, but in his model as well, ATM activation doesn't occur fully. So this is where we left it. And so we were really intrigued about the DISCAN4 because it's a protein that is usually expressed at the two cell-like state. And there's really not much. I mean, it's known to respond to a lot of stimuli, to a lot of genome integrity issues, but it's really unclear what it does. So we really wanted to know if that was the key molecule. So it turned out that to be a very tricky thing to do in mouse because there's multiple copies of DISCAN4. Because multiple copies of DISCAN4, they all seem to do the same thing, and they span a large region of the genome, which is really repetitive. So dealing with this was really tricky. But Marta is a phenomenal postdoc. So she went on and deleted every single one of them. Took multiple rounds of PCR and screening, but she was successful at creating eventually a clone that is DISCAN4 null. And at this point, she could do this, the key experiment, which was to delete TRF2 in the absence of DISCAN4. And what she could see was that you can see end-to-end chromosomal fusion, which was good. So we were right, DISCAN4 was indeed suppressing the DNA damage response. We also saw DNA damage, but the level of fusion is still relatively low. So in the somatic cells, I show you these massive spaghetti-like fusions. In this case, there was hardly any long trains of telomeres fused together. So this told us that DISCAN4 is doing something interesting, and Marta is going to start a lab in the next few months. So she's going to be taking on this project. But from our point of view, and from Marta as well, it meant that there was something that we were missing. So we went back to the drawing table, and we decided to do a genome-wide screen, a really deep genome-wide screen, to find anything that could selectively kill TRF2 knockout cells. So the rationale for this experiment is as follows. So we thought that in TRF2 knockout cells, which again are happy and can grow, there is something that protects them. So there's something that allows them to suppress the DNA damage response. It could be DISCAN4 and additional factors. So by doing a genome-wide screen, we should be able to knock out genes that selectively kill TRF2 knockout cells and not TRF2 proficient cells. So this is clearly, there is a lot of assumption in this approach, but we thought this is the best that we could do. So we did it, and the screen worked well because we picked some genes that are here highlighted in yellow that we previously identified as being essential to prevent end-to-end chromosomal fusion in this particular setting. I'm not going to repeat it. This is in the paper. But one of the things that struck us immediately was a lot of these SMUG proteins. There was a lot of them, and I didn't know much about them, but they're all part of the nonsense mediated decay pathway. And so since we found so many of the subunits, we thought maybe we are onto something. Maybe this nonsense mediated decay pathway somehow is involved in telomere protection in this particular setting. So to test this, what we did, and just probably I'm the least qualified person to tell you something about the NMD pathway, but basically it's an RNA surveillance pathway that can recognize premature stop codon and degrade them. And it's a cascade of events where you have an apical kinase MAC1 that is activated when there is a premature stop codon, and it triggers the activation of a set of enzymes that can cleave RNA in different fashion. But I'm not going to go into specific details because we think in this particular case, all of the proteins that we found have the same phenotype. So we don't think there is a specific function for each individual subunit. Okay, so the very first experiment was to delete them and try to see if they were indeed synthetically lethal with TRF2. So the first good thing for us was that all of the subunits that we were able to knock out, basically every subunit that we did, they're all viable. So here you can see Western blot, and you can see some example of colony formation. They grow fine. If anything, they grow better than the controls in all the cases. And what was really exciting was when we co-depleted, we deplete TRF2 in this setting, now the cells were completely wiped off. So there was hardly anything growing in TRF2 deleted cells in the absence of these NMD factors. So why do they die? So the most obvious thing was looking at telomeres. And again, I don't know if you can appreciate this, but you should be able to see that while in the single knockout, so in the NMD knockout, chromosomes are fairly fine. So that's completely fine, actually. In the TRF2 case, you start having a lot of trains, so where you can see multiple fusions of chromosome all stuck together. So this is now much greater than what we saw in the case of ZSCAN4, and in the case of pretty much anything that we studied so far. So clearly, NMD activity is somehow really, really important for survival of these cells. So this was nice. We were onto something, but what does NMD do? And so there was a couple of hypotheses. So the easiest one to wrap my head around was the canonical function of NMD. So NMD is able to control specific isoform and as well as globally RNA stability. And so you could imagine that in the absence of NMD, you might have less expression of a protective gene that is involved in protective telomeres, or maybe all of a sudden an expression of an isoform of a DNA damage response gene that now can recognize the damage and lead to end-to-end chromosomal fusions. The second option was a non-canonical function of NMD. This was suggested by the Lingner lab when they discovered a long RNA called Terra, which is localized, it's transcribed and localized at telomeres. And NMD seems to regulate the amount of Terra bound to DNA. So we worked a lot on this side, but all the data that we generated is negative, so I'm not going to show you any of that, and I'm just going to say that it doesn't seem that in our case, the non-canonical function NMD is important. So what about the canonical function? And for these, so we teamed up with Bob Yog at NIH, who's an expert on NMD. So he gave us a lot of suggestion on how to find potential targets for NMD. So what we did was, what we ended up doing was to do very deep RNA sequencing, total RNA sequencing, where we're trying to capture any single isoform presence in wild type cells or SMAC5, SMAC6, or UPF1 knockout clones. And we did it multiple clones, multiple times. And then we looked for any weird transcript that was indicating that the NMD was targeting these transcripts. And then we combined basically what is in common with all of them. So we were left with around 300 transcripts that were affected the moment you didn't have NMD. And so one thing that is important here to mention is that NMD is known to be essential in somatic cells. And the fact that the NMD knockout mouse embryonic stem cells were viable could have suggested that NMD was maybe not a thing in ES cells, where NMD is clearly a thing because you can see all these events coming up. Moreover, when you look at these common targets, they all fit what is known about NMD activity. So mainly splicing factors, RNA regulating enzymes, which are known to be really good targets for the NMD. And I'll show you one example because I had a lot of fun looking at this transcript. So you can look at the transcript and see what happens when you don't have NMD. So this is a good example of a known NMD target called the SRF7. Oh my gosh, sorry. Okay, so here I don't see anything, so I'm gonna go here. So what you see in this case is the control here. So these are NMD proficient cells, and this is 3 different genotype, all NMD deficient. And this is SMAX5 for some reason is the one that has the cleanest phenotype, where all of a sudden you have this cryptic intron that is retained. And you can see that you barely see this event in the control, but this is really prevalent in NMD knockouts. And this is exactly what you see. So it means that in normal cells, these transcripts are normally wiped off, degraded, while if you don't have NMD, this is maintained. And this is in the case of SRF7 and other splicing factor is known to be a regulating function. So in a way to control the amount of splicing in cells. Okay, and you can do this looking at the old transcript, or independently we designed qPCR primers that can detect specifically this isoform. And you can see that when you don't have UPF1 or SMAC6, you can detect them really nicely. So all of this is telling you that we can detect weird transcripts in NMD and they behave the way you expected them. Okay, so but SRF7 was not really the most exciting gene for me. So I went past the known RNA factors and tried to see if there was anything else that could make some sense. And immediately it took a genius to look at geo terms and seeing that there was a lot of telomere associated proteins that came out in these core genes that were deregulated. And among them, the top one was TRF1, top one in terms of the amount of weird transcripts that were retrieved. So this is what you see for TRF1. So again, sorry, I'm coming here because otherwise I cannot see. So in control cells, TRF1, it behaves fairly well. So this is, we're looking at the terminal portion of TRF1, sorry, as 10 exons, and you can see that they all splice regularly. However, in the absence of UPF1, SMAC6, and I'm not going to show you, but even in the other one, you have a very frequent event that skips completely exon 8, where you can see exon 8 is skipped in roughly 50% of the transcript, so quite prevalent. And so this was interesting and potentially relevant because when we looked at the protein, so what would potentially do this new splice variant, was quite intriguing to think that we were maybe generating a dominant negative allele. So the top here is the way a normal TRF1 would look like in the exon 7, 8, 9, 10. So if there is no skipping of exon 8, this will produce a fully functional TRF1 that has a DNA binding domain where it can bind telomeric DNA. However, if you skip exon 8, this will create a premature stop codon that will lead to a truncation presumably should lead to a truncation product that would not have a DNA binding domain. And the interesting thing is TRF1, like TRF2, it's a homodimer. So there is always dimerization between molecules. And so if you add this delta and exon 8 TRF1, this could potentially act as a dominant negative because it would bind to the other functional allele and lead to a reduced level of TRF1 at telomeres. At least that was the prediction. So the first indication that that could actually be the case was just looking at the proteins. And you can start seeing that in the case here, we have SMAC6, we have accumulation of a shorter fragment of TRF1. By sequencing the RNA, we really think that that's exactly what we predicted to be here. So the delta exon 8. And then we did a lot of experiments. I'm going to not show all of them, but I'm just going to show you that, to me, what is the strongest is we reasoned that if indeed the main phenotype of the NMD knockouts losing protection of telomeres was due to this dominant negative TRF1 allele, overexpression of a wild type TRF1 should overcompensate the system and lead to full protection. And this is exactly what happened. So what you can see here is in the absence of NMDs, you can do this in all the different background. When you delete TRF2, you can see end-to-end chromosomal fusions. But if you overexpress TRF1, that doesn't happen. So the cells are fine, there's very few fusions, and the cells are viable, suggesting that the reason why these cells dying is because of TRF1, dominant negative allele. Okay, so you're not going to be able to see much of this, but basically this is the way we currently thinking about stem cells. And so what we think is happening is basically in somatic cells, deleting TRF2 leads to ATM activation, and this is end of the story for the cells because ATM will lead to end-to-end chromosomal fusions. However, in mouse embryonic stem cells, for genes such as DISCAN4, the ATM pathway is dampened. So when you delete TRF2, this doesn't really elicit a full-blown DNA damage response. So the cells don't die and survive. However, if we mess with the NMD, we have less TRF1, and this, and again, I didn't show you the data, but this will lead to a DNA damage response that is mainly ATR dependent, leading to DNA damage activation and fusion when it's co-depleted with TRF2. Okay, so we think we have an explanation for this, and you might, I mean, one thing that we started to think now, okay, do we care? Okay, this is clearly a very peculiar system, these embryonic stem cells, where we have to delete so many things, so does it mean anything? I like to think that it does. Of course, I'd like to think that it does. But so one of the reasons why I think it does, or at least evidence that I found that I think it does, is one of them is DISCAN4. So this molecule here that seems to be suppressing ATM. This is found overexpressed in some cancers. So again, Marta is collecting cancer cells in which DISCAN4 is overexpressed, and one of her projects will be to understand whether in these cells DISCAN4 provides a telomere protection. But more generally for the NMD, I think it could be really interesting to see if this, I mean, since there is a number of telomere-associated protein, whether that's really a key function of the NMD pathway. So NMD, again, is interesting from a cancer point of view. For instance, one idea is that you can target the NMD to increase instability, and so leading to better therapy, immunotherapy. And could some of this be mediated by telomere dysfunction? We don't know. So, but we are doing now, we're making somatic cells in which we can delete the NMD to see if there is any signs of telomere dysfunction, as well as looking at the ES cells that have NMD mutation to see if they have any defect in telomere biology. So again, we think that understanding how stem cells do this weird protection, we might be understanding something about some cancer cells deal with telomere dysfunction. So at this point, I'm going to be moving on to the second part of the story. So if anybody has questions, you can keep them or you can ask them as you want. I can, yes.
I was wondering if you had any insight on what determines which exons may be skipped, because you mentioned like that skip.
From
what could be a determinant factor which exons could be skipped versus not?
So that's a really good question. I do not know the answer. Meaning it could be, so basically the way I think about it. So these events is happening all the time, and if you're in an NMD proficient setting, the NMD will clear off those transcripts, at least in ES cells. And you do see, so if you look at enough sequences in the control, you will start seeing some evidence of that. In somatic cells, we don't see that. So, and I don't know, it could be, so there's different, so one option could be simply the output. So there's more transcript for TRF1, so there's more chance of skipping that exon because, I don't know, maybe the splicing events are not very efficient. Again, there's people more qualified than me in this audience to address that question. But it could also be a regulatory. So for instance, ES cells are known to elongate telomeres really a lot early during embryogenesis. So maybe that could be one way to create a dominant negative allele of TRF1 that could, so TRF1 is a known negative regulator of telomerase, so it could lead to higher level of telomerase. That's just pure speculation. That's what we're trying to do now. Yeah.
It was like, is TRF1, TRF2 double mutant also?
Yes.
Sufficient?
Yes. TRF1, TRF2. It's phenocopies. I mean, it's if anything, is a little bit better. So is even more penetrant. There's more fusions. We never in ES cells, we never get to the point of somatic cells, and I think. I don't know if I don't know if I want to delete half of the genome to get there, but I don't know if we will ever get there, meaning that it could be a cell cycle. There might be a limit to the amount of fusions you could have in these cells. Yeah. Yep.
So they generated mouse brain control, correct? So do you have any data, like similar data on human brain itself or at least?
Yeah, so we don't. So one of the drawback of NIH is that it's really challenging to work on human ES cells. We can't. So we should do IPS. We didn't do it. We didn't do it. Yeah. But that's on the pipeline. So we had actually a collaboration with Luis Batista, Washu, and he tried to do some TRF2 knockout cells. It was not successful. And I don't know if that's because they're dead or because it was not successful. So one of the things that we started to do now, it's DTAGs. So we DTAG, at least in mouse ES cells, every single telomere component. So we're gonna try to do that maybe in IPS. It could be nice. Yeah. Yep. Oh.
Do you see the teams? We do see the teams. Yeah, one.
Yeah. Yes. Sorry, I mean, we didn't do chip that we should do by even by IF. It's really dimmer, if it makes any sense.
So heterodyne combined with the wild type.
So you could, so you're gonna have a mixture, right? So let's say 50-50, you're gonna have some homodimers that are completely fine. So there's some TRF1, but all the heterodimer with, and this we also showed, and again, I maybe should have included those, but if you overexpress that delta exon 8, you're gonna achieve the same phenotype. And I don't know, like, if this is conserved in human, it would be really interesting. So far, I haven't seen evidence for that. But, yeah. Rishi, yep. End-to-end chromosome fusion in.
Yeah.
So with these cells, it's difficult to do. So ES cells are quite fast for our, at least for our time scale. So they are roughly 12 to 14 hours division time. And TRF2 deletion done this way, it takes longer. So basically, by the time we put tamoxifen, we have to wait 72 hours at least. So it's really difficult to pinpoint when. But again, now we are doing it with DTAG, and that's much faster. In 4 hours, we can get a deletion. We don't have double DTAG and MD TRF2. But it looks like that. So most of the fusions, again, so if you look at the fusions, they're all G1 fusions. So they're all chromosome fusion. You don't see any chromatic fusion. Yeah, that's a really good question. And I know we did this sperm, but I don't remember the answer. I should. I really don't remember. Yeah, I don't remember. Sorry. Yeah. Okay. So I'm gonna move to the second part. Okay, the second part is. So there's a couple of things here that I think if you lose me for the rest of the talk. So please do stop me, okay? I'll do my best, but do stop. Okay, so this has to do with the way cancer cells elongate the telomeres. Told you at the beginning, so there is telomerase. Telomerase accounts for roughly 85 to 90% of the way telomerase is maintained in cancer cells. And it's clearly the way germ cells in our body maintain telomeres. And one thing that I should have mentioned before, most of the cells in our body do not express telomerase, so that's why cancer cells need to engage a telomere engagement mechanism. And I think telomerase is quite well understood, so we know the enzyme. There's still some questions, so we don't know precisely how it's recognizing the telomeres, so it's a crowded space, and you have to find the very ends. But for the most part, we know, and we also know the way it's induced in cancer. There are mutations in the promoter that leads to telomerase reactivation. But there is a 15% of tumors, and these tumors are not equally distributed. So for instance, also sarcoma is really predominant, this type of telomere elongation, that do not express telomerase, and they still maintain the telomeres. And they use this in a alternative lengthening of telomere pathway, which is a way of saying that there's still some things that we don't understand. And so the way I think the field thinks about this is basically a break-induced replication mechanism, where you have a telomere invading, so potentially a short telomere invading a longer telomere, and using this template as is template, leading to a net gain of new telomeres. So this is a homologous recombination-based type of elongation. But there's still a lot of questions here that are unclear. So it's unclear, and does it happen normally? How does it start? Why not all cells do it? And you can probably mention a lot more. But what I'm going to try to address here is some even more fundamental questions that we don't know about alt cells. And the first one, and the way we started doing this project, is something that we did a while back. And so Taylor Lowe was a student in the lab when I was in San Diego, and we had this maybe not, I thought maybe crazy idea at the moment, but it turns out to be right, that we might be able to distinguish alt cells from non-alt cells just by looking at single-stranded DNA at telomeres. So what Taylor did was to do a native, so we call it single-strand TLOC, so basically a native fish, or in a different cell, clearly, denaturing fish for telomerase. So this is the same probe, just done under native or denaturing condition. If you do it in denaturing condition, all the cells that we ever analyze have telomeric DNA, as long as they are mammalians. But when you look at the native fish, only alt cells show some signal. So you can see, hopefully you can see this in these red dots. They're distributed. There's not always the same amount, but there is a signal in the alt cells, and there's never any signal in all the alt negative cells that we ever tested. So my interpretation of this is that there is a lot of single-stranded DNA in alt telomeres, but you could argue, and you would be right to argue, well, maybe PNA is we're using PNA probes, they might be very invasive. There could be many artifacts in this. But this it's a really strong phenotype. We can use it in different cell types. We use it in dog osteosarcoma cell line, and we can distinguish alt and non-alt cells. So there's something here. And I was intrigued about this. So why would there be single-stranded DNA? And this really got me thinking about telomeric DNA. There's still a lot that we don't know about telomeric DNA in alt cells, period. So we wanted to study this, but it's complicated because telomeres are repetitive sequence, perfect repeats. So if you're talking about alt cells, you have up to 50 kb of perfect TTHG repeats. There might be a couple of variants here and there, but usually. So they're difficult to map. So if you do a chip for RPA, it's not going to tell you anything because you're going to get TTHG, you don't know where it's coming from. So, but I was lucky when I started at NIH, my boss and my colleague is Andrew Nussbaum, who developed a method to study DNA damage by isolating the DNA. So this is called NSeq, and this is the first iteration of the NSeq that they did. So the basic idea is very simple. You take your cells, you embed them in agarose, and then, so you do this to prevent introducing breaks, and then you polish the end and add adapters. And if there is a break, the adapters ligate to the double-strand break, and then you can sequence it. Since the sequence is always going to be directional, if you have a double-strand break, you're going to get two reads, one on the bottom strand of the break, one on the top strand of the break. So if there is a double-strand break, that's what you see. And this works really well. Andre published a bunch of papers on this. So we thought, could we use it to understand something about telomeres? And so before I go into alt, I have to just introduce the method. So I'll give you a couple of slides. And this is the moment that if you lose me now, you lose me forever. So if you want to sleep, it's a good moment too. Okay, sorry. Okay, so Benura in the lab decided to try and use NSeq to study telomeres. So telomeres are one-ended break. Right? So there's basically, it's a double-strand break, but it doesn't have the other portion. So if everything works fantastic and you take your cells and embed them in agarose and ligate the oligo, the oligo is gonna ligate this here, and the only strand that it should always get is this bottom strand, which in our case is the C-rich strand. It doesn't matter, but it's always gonna be the same at every single chromosome end. So in other words, there's nothing on this side, so you should never get any sequence corresponding to the G-rich strand. So this, if it works. Of course, if there is a break in the middle of the telomere, then you're gonna get everything else. So the first experiment was just to look at the sequence and can we only detect the right strand? And this was indeed the case. So Benura did a ton of, I mean, just showing you a very small fraction of what we did. But in this case, we looked at mouse, human cells. These are the primary cells, so cells that have telomeres. And in all the cases, you only see the right strand, so the bottom strand, suggesting that we are really indeed detecting the very end. We recently started looking at fish and dogs, and everything always works the same way. Okay, so the next thing you can do now, you have all these reads. These are short Illumina reads, so we have millions of reads. We can pull them out and ask, okay, what is the sequence at the very end? And this was exciting in many ways because we saw that the sequence is not random. So most of the telomeres in your body always end with the same sequence, which is ATC. This is true in ELA, but again, it's true for most of the cells that we checked. This is exciting, but we were not the first to find this because 15 years ago, someone already showed this using a more complicated way. But in a way, this was good for us because it confirmed that we probably are detecting the very ends. And in addition, the method that we're using in this paper, it's really convoluted, so it's really difficult to map, let's say, long telomeres like mouse or alt cells. So this is an improvement over what was done before. And we did a lot of controls to make sure that we look at the end. I'm going to show you just one. And this is basically, if we are right that we detect in the very end, since the end is not random, we should be able in a plug to randomize it. So you can take your plug, and you can put an enzyme called T7 hexanuclease that is going to chew the end, and it's going to do it in a random way. And then you can run your end-seq. If everything works fine, you should have a different sequence. And indeed, that's what we see. So if you don't treat with T7, you get ATC. But in the same plug that now is being treated with T7, you get roughly a randomized, almost perfectly randomized end. So this is telling us that we are indeed detecting the ends. Okay. So now we can go back to ALT. Okay, with ALT, we had two questions. So the one question, which was simple, was like simply saying, is the end, since they use this weird recombination method, could it be that now the ends are not ATC anymore? Could it be that now the sequence is completely different? The answer was no. So the sequence is still the same, it's still ATC. Okay, but the more pressing question is, can we detect single-stranded DNA? And if it's there, can we understand where it comes from? Okay, so that was the main reason why we did this. And here I'm just giving you some of the speculative molecules that could give rise to this single-stranded DNA, if there is a single-stranded DNA. So if you ask anybody working on the telomere field, they would all bet on this. I mean, I think the vast majority on these structures called C circles. So alt cells are known to have been described to have this extra chromosomal C-circular DNA, which is partially single-stranded and partially double-stranded. So this could be a source of single-stranded DNA. They could have gaps, like just gaps. They could be R loops due to terra, as I showed you before. There could be invasion, the D loops, so due to the recombination pathway, or there could be internal loops. So these are some of them. Maybe I'm missing something, but this is the kind of things that we're thinking of. But again, all of these would only make sense if we can detect single-stranded DNA. So to detect single-stranded DNA, what we decided to do was to use a iteration of N-seq, which is called S1 N-seq. So the idea is very simple. It's similar to N-seq. So the only difference before you do your ligation, you incubate your plugs with S1 nuclease that cuts single-stranded DNA and not double-stranded DNA. So it will convert any gap, any single-stranded DNA, and it will target all the structure that I told you before into a double-stranded DNA, and now you ligate. This is going to have an important implication for telomeres, because so if you don't have any internal single-stranded DNA, S1 is going to be exactly as N6. So basically, when you do S1, the only sequence that you will always get is ATCC, because you're not creating any new breaks in the middle, right? Okay, however, in any of these conditions, so C circles, all of them, there's now some single and we do this, we only get always the C strand. Almost completely, some of you might pick up a slightly green here, which is, we don't know, but it could be these cells have long telomeres, so there might be some replication intermediate that might be converted to double-stranded break. But basically, for the most part, we only detect the C. So there's no single-stranded DNA, and that fits with our data. Now, when we look at ALT-positive cells, and we looked at the bunch, in addition to the one that I'm showing now, in all of the cases, we see, quite a lot of reads coming from the G strand. The ratio is almost 50%, so that's a lot of breaks. So it would mean since any single telomere will have one read that comes from the very end, plus anything that comes from inside. This tells us that there is almost a 1-to-1 ratio between, or even more actually, between the ends and the internal gaps. So there's quite a lot of single-stranded DNA, which I think it's remarkable, because I mean, at least I always was under the assumption that having a bunch of single-stranded DNA in the genome is not very good for proliferation, but these cells grow really, really well. Okay, so we have these reads, and now where are they coming from? So one of the first things we did was just to look, okay, what is the sequence around these reads? Can this tell us anything? Of course, the sequence is TTHG, but what is it? And so the boundary is not random again. So we always have this RSC or G, depending on which of the strands C you're looking at, but the boundary is, it's always the same. So you have a GG or CC. And again, this is different from what we saw at the end, where you had this ATC. Unfortunately, this doesn't tell us anything, because all of the structures that I mentioned before, they could all have these structures. Nevertheless, it tells us that there is single-stranded DNA, and there seems to be a motif around the single-stranded DNA. Okay, so how do we understand what is the main source of this single-stranded DNA? So the thing that we thought of doing was, well, there is a distinction between some of these molecules. So in some cases, we have 5' ends. So in the C circles, we have, 5' ends. If there is gaps, there's clearly going to be 5' ends. All the other structures is that they don't have discontinuity in the helix. So there's going to, I mean, they're unpaired, but there's no breakage itself. So there's no free additional 5' end. So the prediction with this would be that these guys should be T7 positive, while all the other guys should be T7 insensitive. And doing this, we could at the very least distinguish these two main categories. So that's what we did. So we did the T7 treatment the same way that I showed you before on the very end. And just as a reminder, so these plugs, we take these plugs, and if you just do an end-seq, so we just look at the end without cutting the single-stranded DNA, and you do T7, you can randomize the ends. So the very same plug now, if you do S1, and then, sorry, T7 and then S1, you cannot randomize it in any way. So this is really suggesting that these guys don't seem to be the main source of this single-stranded DNA. So in my opinion, based on this data, the C circles do not seem the main source of single-stranded that we can detect in our assay. There doesn't seem to be really single-stranded DNA gaps, but these are some of the structures that could give rise to this. Most of you don't work on telomeres, but if you do tell this to people working on ALT, they all get very wired up and they say, what are you talking about? So I've been thinking of ways to sort of support or not support that, to see if there is any other way that we can detect this. So the way that we came up with, it's not the best, but it's one way, is like we try to do in situ amplification. So basically the basic idea here is that we have cleanup, and if there is 3' or 5', if there is a 3', cleanup should be able to add biotinylated UTP in cells that have been fixed. This again is people do this all the time. However, if in all the other conditions, so this would apply to the C circles, it would apply to the gaps. But in the other condition where instead there is no gaps, this shouldn't work. And the only way to make this work would be providing a primer for annealing and then letting cleanup do its job. So we did this in many different cells. I'll just show you one example with U2S. But what you see here, if we put in the very first condition, so we just clean up and bio UTP, we see absolutely nothing. However, if we have cleanup plus a primer, now we can start to see a product. So again, this to me suggests, and of course, if you don't put cleanup, you don't get anything. To me, it makes me more confident in saying that I don't think these are the source of single-stranded DNA, but some of these guys must be, okay? And I'm sure people would still be upset about it, but that's what we have. Okay, so finally, I'll just give you two quick data trying to tease out between R loops and ARA DNA intermediate. So for R loops, what we did was what most of the people that study R loops, we overexpress RNase H. With the idea is that if these structures are R loops, we should be able to reduce their levels by just overexpressing RNase H. And this was not the case. So yeah, I'm just showing you the amount of single-stranded DNA that we can detect by S1-Sig. And there's really no difference whether we have a catalytically dead or a functional RNase H. There's absolutely no difference. And this is the same that is in the wild type. So we don't think it's R loops. But we do think, or at least our data suggests that they're really linked with the ALT reaction itself. So we have different ways to suppress ALT in ALT-positive cells. One way is to deplete Bloom, the BTR component, the helicase part of the BTR component. We published this a while back. And if you don't have Bloom, you basically have no ALT activity. And in this case, you can massively reduce the amount of single-stranded DNA that you see. And the same is true if you overexpress ATRX. U2S cells don't have ATRX. And if you re-express ATRX, the cells stop doing ALT very efficiently. In both cases, you see a significant reduction in single-stranded DNA. It's not complete, but it's significant. So basically, in conclusion, I personally think that what is happening is either we're detecting D loops, so intermediate of the recombination itself, or the model that I like the most, but I don't know if it's difficult to prove, is that maybe these reactions, since again, you have to think that these are 50 kb of perfectly matched TTA GGG repeats. Once you have the invasion, once you resolve this, it might be very difficult for cells to put things back completely. So there's a lot of chance of having basically loops created. In addition to this, these cells have a defect in ATRX and DAX, which are required to deposit at H3.3. So the chromatin might be also altered. So all of this might give you a chance of creating some medium, small, we don't know, big size loops. And I think it's interesting from, there's a lot of things that we could try and target specifically these cells. So I can stop here, or I can give you 2 slides on really quick. I mean, there's really no data in this, but just to where we're going with this idea. So one of the implications of what I just showed you is that the single-stranded DNA that we detect is really linked to the ALT activity. So we can use this then to try and find regulators of ALT. So we set up a screen with Gianluca Pecoraro at NIH, who does basically phenotypic screens. And so in this screen, basically, what we do is, I'll go right to this. So I'll just show you just some controls. So we can transfect in U2S cells guides against genes that are required for ALT, so Bloom, PML, and RMI. Again, we showed this before. And in all of these cases, you get a reduction in the amount of single-stranded DNA, or we can even increase the amount of single-stranded DNA by expressing a gene called TZAP that we showed before, that it can enhance the ALT phenotype. So these are our controls, everything's working. So then we went and do a massive, not massive, but a semi-big screen for us. So we made a custom library with 1,200 genes. So these genes include DNA damage protein, epigenetic genes, different factors that we think could be involved in the ALT process. And then we do this arrayed in a 384 web format. And then we look at single-stranded DNA. And when we do this, things work well. So we can detect at the bottom, like we can detect PML and Bloom, which are genes that are involved in ALT. And at the top, we can detect genes that have been shown before to be suppressing out, like FANCAM and its associated protein. So, and hopefully, as you might see, we have a bunch of other proteins that now we are going through. We validated a couple. It's still really early, but I think we have I think we have a way to at least trying to put new pieces in the puzzle to understand how ALT functions, and hopefully, and that's going to give us good result and potentially ways to target these cells selectively. So with this, I'm going to stop, and I'm just going to thank. I mean, this is the most important slide. So hopefully I did a good job of mentioning. So Marta is a postdoc that did all the work on stem cells. Benura did all the work on stem cells, and she was helped by Gianna, the student in the lab. And we have a lot of collaborators. So I'll just mention Kyle and Travis that we're working together on the final part of the screen that we're working on. And hopefully I thank everybody else. And finally, please rate this seminar. Yeah. Well, no, okay. Yeah. Okay, I'll take any questions. Yeah. Thank you. Yeah.
Does it like increase decay of density, and does it change like ratio of TRF and
TRFs, I should know this. I think so. I mean, the only paper that I think of is from a young Calcider, and I think they did show that. It's tricky, the nucleosome, unless you do ataxic as we were discussing before, but by classical micrococcal nucleases, the resolution is not that high. So I don't remember specifically somebody doing ataxic, for instance, and I'm sure people did it, I just don't know. But there are backup pathways, so the ASF1 can, for instance, deposit also. So I don't know. Long way to tell you that. I don't know. Yeah. Oh, sorry. Yeah. 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 So the resolution is fairly bad, right? So we don't know. So you're gonna basically. So let's pretend that there is 10 single-stranded DNA portion per telomere. 10, 7, they're gonna give us the same exact information. So it could be that there are loops, but they're just not as frequent as other type of invasion. So there might be a reduction, but we just cannot. The resolution of this is fairly bad. Yeah. Yeah, it could be that the R loop starts it and then stabilized by D loop for sure. Yeah, but in general, any negative result is tricky to. It could be the RNAs. I mean, even if you get rid of the terra in that setting, the loop stays. That could be another option.
From Hassan Sabawi. Hi, nice presentation. In the NFT work, how to account for differences in cell cycle checkpoints between ES and
Yeah, so that's.
Can you address this question because they can't hear?
Oh, okay. So shall I repeat the question?
And also,
okay, so basically the question is whether how do I account for difference in cell cycle and differentiation status in, let's say, the NMD or all the others. I didn't show you, but clearly we look at stem less. So NMD, all the NMD knockout, they're still stem cells. They didn't differentiate. Cell cycle grossly, they are completely fine. So there's no difference in differentiation. Between stem cells and differentiated cells, clearly there is a difference in cell cycle. But we don't think that's the key for the protection for many reasons. We publish some of them, but basically you can get normals in joining in those settings.
Right, so you get non-normals in joining elsewhere.
Even at telomeres. So if you, let's say, for instance, in the NMD TRF2, we get normals in joining. Actually, I mean, we do get normals in joining the setting. The cells are still stem cells, so they can do normal. Yeah. To do it, yeah. And the other question was on heterochromatin. We didn't do it. We should do it. We didn't look at that. Yeah. Yeah.
Did not see biological fluctuation without that.
We do not see in U2S cells. No, we do not.
You have a standing network, you should have,
If we have a strand embedded, we should see that. Yeah, depend on the displacement, strand displacement activity. And I believe that we didn't use a strand displacement polymerase, but we should do that. It would be nice to do like a phi29 in that setting, right? Yeah, but that's a good point. Yeah.
The loops that they, any of them replication forms associated, or you think it's the end?
Okay, the loops you mean in the S1 or in the, so in the S1, this should be, because I mean, if there was a, well, I don't know, so sorry, I know you didn't ask that, but I'm thinking of a reverse fork, which I know is not what you asked, we asked, but a reverse fork would look the same as a telomere in a regular NSIC. But replication intermediate, yes, they should be detected by S1. So we did try TRF1 early on. So TRF1 deletion should lead to more problem replicating telomeres. We didn't do it thoroughly enough, but we didn't see a lot of increased S1 signal. So I don't know if it's too transient, if it's too, I don't know. I don't know why it wouldn't be.
More likely.
In our case, yes, because it's difficult to imagine that. So ELA would have a lot of replicating events. So we only see them so that's why I was pointing to the S1 ELA long T, which is, you see a little bit of reads from the other direction when you do S1, but it's really, really little. So those could be replication, we coat the cell in during replication. But other than that, we don't see any signal for S1. So I don't think so. I mean, again, you're dealing with millions. So this experience is 50 million cells. A lot of reads, the reads that are telomeric, it's a subset. So you probably would need to arrest cell in S phase and stack them there for a long time to see this. I'm sure you could see it, but we didn't do it. Yeah.
So Eros, given that you're sort of demonstrating perhaps that there are no real C circles, which other people that are not involved.
Yeah.
I'm just wondering if the T loops and the internal loops that you're proposing are the main feature blocks and the reason for your signal, if they could give, if the methods, what is it?
Yeah, rolling cycle. Yeah.
It's not an argument, but
Yeah.
the C loop is created
Yeah.
by that polymerase people use to.
Yeah, I don't know. It's difficult to. It is difficult to. So you could, basically, if I was a C-circle aficionado, I could find many reasons why our method doesn't detect it. For instance, it could be that the double-stranded portion is too small, so that would not be detected in our method. So there's ways. The ways. Yeah, no, it could be. It could be. It's difficult. Yeah.
Could also be detecting those.
Could be. It could be. If you draw it, and I tried many times, I could not come away on a way that it would do it. But if you find a way, please tell me, because I'm sure that's going to be a reviewer number 3 question. On the C-circle reaction, so I don't know if anybody has ever sequenced the C-circles. I was thinking about that because I don't not that I know, but that could be an interesting thing. Yeah. I don't think it should though, because I mean, phi29 is super processive. These reactions are done over many hours, so you leave them overnight in. So I don't know. I don't know. Yeah. All right.
Thank you, Eros.
Thank you, everybody. Thank you.